Up to now, we have considered what happens inside of a cell to produce energy if the cell uses glucose and has oxygen. Now we want to take a look at some other situations where perhaps the cell does not have oxygen available to it and or it doesn't have glucose available. First, we're going to take a look at the situation where it does not have oxygen. These situations are referred to as anaerobic. Remember, aerobic was in the presence of oxygen. Anaerobic is in the absence of oxygen. Let's take a look at metabolism without oxygen. Glycolysis can occur in both aerobic and anaerobic environments because recall that glycolysis itself doesn't require oxygen. So it can be in aerobic environments, but it also can occur in anaerobic environments. In glycolysis, one of our input molecules that we required to run glycolysis is NAD because we're going to generate NADH. If we don't have NAD available, we can't continue to run glycolysis. When oxygen is available, that NAD is regenerated during oxidative phosphorylation because remember, we take NADH, we take the hydrogens off of them, and that regenerates our NAD. And that NAD then can be shuttled back out and used to run glycolysis. If we're not running oxidative phosphorylation, the problem we're going to encounter is that NADH will build up and NAD will not be regenerated. And so if that occurs, we need a way to get NAD back to at least be able to keep running glycolysis. There's a couple of major pathways that exist. One is called fermentation. Fermentation is a step that regenerates NAD. That's really the primary purpose of fermentation, is to get back our NAD so we can keep running glycolysis. Glycolysis doesn't give us a lot of energy, but it's the only thing we can do in the absence of oxygen in certain types of cells. But we can't keep doing it if we don't get our NAD back. Fermentation gives us that last step to regenerate NAD so we can keep running glycolysis and getting our small amount of ATP from that. We'll look at 3 different types of fermentation. We have lactic acid fermentation that we see in certain types of cells. For example, muscle cells that don't have enough oxygen will resort to lactic acid fermentation. We also have some yeast that can perform alcohol fermentation. And then when there's some archaea that perform methanogenesis, that essentially create methane. Also, there's other types of cells in the absence of oxygen that can run a different type of pathway known as anaerobic cellular respiration. Anaerobic cellular respiration is distinctly different from fermentation because it allows us to continue to break down the glucose very much like we did in aerobic respiration. So its sole purpose isn't just to regenerate NAD. We will generate a lot of more ATP from anaerobic cellular respiration. We just do it in the absence of oxygen. We'll take a look at these. First, let's look at lactic acid fermentation. Lactic acid fermentation occurs in some muscle cells when there's not enough oxygen for those muscle cells, some mammalian red blood cells, some bacteria will do lactic acid fermentation. For example, the bacteria that we see in yogurt. This shows us that pathway. We did glycolysis, we took glucose and we broke it down into pyruvate, and we generated these 2 NADHs. And as we said a minute ago, if we can't get these NAD back, we can't keep running this reaction. So this fermentation step is really all about getting this NAD back. We take our 2 pyruvates that we had at the end of glycolysis, we convert those to 2 molecules of lactate, but in that fermentation step, we oxidize NADH to NAD, and then we can shuttle this NAD back up to glycolysis. So we don't really get any more energy out of this step, but what we do get is our NAD so we can keep running glycolysis. Lactate dehydrogenase is the enzyme that will catalyze this reaction. In this reaction, we take pyruvate and NADH, we get lactate and NAD. When we perform this lactic acid fermentation, we're not going to get any more ATP. So all we're going to get out of this is we're going to take 1 molecule of glucose and we're going to get our 2 net ATP that we got from glycolysis. So it's not a very energy efficient mechanism, but it does allow muscles to continue to work in the absence of oxygen rather than just completely shutting down. Let's take a look at this video that talks a little bit about lactic acid formation in muscle cells.
It's one of those basic ideas you hear scientists repeating all the time. Correlation does not imply causation. Just because two things happen together doesn't mean one caused the other. Well, back in the 1920s, they ignored their own advice, and the result was a major misconception that's still around today. Because despite what you probably were taught in high school, lactic acid does not make your muscles hurt when you exercise. In fact, it might actually help. It all started around 1920 with a German biochemist named Otto Meyerhoff. He helped figure out many of the steps of glycolysis, which is the series of chemical reactions that cells use to make energy from glucose, or sugar. He also kind of fit the mad scientist stereotype. If you've ever seen a scientist in a movie electrifying disembodied frog legs, well, Meyerhoff did that in real life. He took frog legs, just the leg parts, and he gave them electric shocks, making them jump and twitch. But after a while, they stopped jumping, and Meyerhoff found that the legs were full of lactic acid, or more accurately, lactate, which is lactic acid minus a proton. He and another scientist, Archibald Hill, correctly figured out the steps of glycolysis that led to higher levels of lactate in the muscles. But Meyerhoff also assumed that the lactate buildup contributed to muscle fatigue, stopping the frog legs from jumping around. When you exercise, your body uses up its main source of energy, ATP, within a few seconds. Then it starts to make more, using a bunch of different chemical reactions. And during a hard workout, your muscles start to rely more heavily on reactions that also produce more lactate. When you exercise, the pH in your muscles goes down, meaning they become more acidic. That's called, appropriately, muscle acidosis. The drop in pH may be part of what causes the famous burn while you're exercising. There was a clear correlation between the lactate buildup and acidity in the muscles, and Meyerhoff assumed causation too, that lactate was responsible for lowering the pH in muscles. This became a generally accepted hypothesis, and you still see it all over the place today. Unfortunately, as we all know, assuming causation can be risky, and in this case, Meyerhoff was wrong. Even though the lactate idea was well accepted, there wasn't much empirical evidence for it. And over the years, researchers started to question whether it was accurate. Around the second half of the 20th century, they started to pick apart what was really going on. One important clue was the fact that unlike lactic acid, lactate isn't an acid. That missing proton means it's a base. It accepts rather than donates protons. Meyerhoff's idea assumed the reaction that produced lactate also produced a proton at the same time, making the muscles more acidic overall. Turns out, it doesn't. The series of reactions that generate more ATP in the muscles do produce extra protons, which lowers the pH in your muscles, but the extra protons don't come from the step that generates lactate. And in fact, lactate bonds with some of those protons, making the muscles less acidic. Lactate also isn't involved in delayed onset muscle soreness, the pain you feel a day or two after a hard workout. Scientists think that comes from microscopic tears in your muscles. Although they are still investigating that, it's weird that we don't 100% know, but we don't. So despite what you hear from personal trainers everywhere and sometimes even see in textbooks, there's no reason to be so hard on lactate. It's just trying to help. Also, a side note here, you're going to want to tell people about this when they say, the lactic acid. Do it, but try to be in the least pedantic way possible. We have to be accurate, but don't be condescending.
The second type of fermentation is alcohol fermentation. This occurs in some yeast species. It involves two reactions that occur after glycolysis. We still perform glycolysis and we still get our 2 ATP from glycolysis. At the end of glycolysis, recall that we have pyruvate. So our first step in alcohol fermentation is to take pyruvate and to remove carbon dioxide and create 2-acetaldehyde. Acetaldehyde is a 2-carbon compound, and we've extracted CO2. That step is catalyzed by pyruvate decarboxylase. Our second step takes aldehyde and NADH and creates ethanol and NAD. Remember, our real goal here was to regenerate this NAD so we can send NAD back up to glycolysis. So once again, our primary step here was to get that NAD back. The only ATP we've gained throughout this whole process was 2 ATP. This step is catalyzed by alcohol dehydrogenase. Here we'll take a look at a video about fermentation.
Can
Can you name a type of fungus that is important in the diet of humans? I bet most of you thought mushrooms. However, there is a fungus that is arguably more common in human food. Any idea what it is? In this video, we're going to look at anaerobic respiration by yeast and how it can be harnessed to produce food. Yeast is a type of fungus. Like all living things, it respires. Yeast can respire anaerobically, so without oxygen. Another word for this is fermentation. When yeast respires anaerobically, this is what happens. The yeast uses glucose as a fuel to produce carbon dioxide, ethanol, or alcohol, and energy. The energy that is produced by fermentation is useful to the yeast, as the yeast then uses this for metabolic processes such as growth. However, the carbon dioxide and ethanol are waste products for the yeast, but useful products for humans. One way humans use the fact that yeast fermentation produces carbon dioxide is in bread making. Yeast is mixed with flour, sugar, salt, and water to make dough. The yeast then respires anaerobically, producing carbon dioxide. Bubbles of carbon dioxide are trapped in the dough of the bread. This makes the bread rise, resulting in a lovely soft, bouncy loaf of bread. Yum! Why do you think sugar is added to the dough? Pause the video to have a think about this. Sugar is added because it is the fuel for respiration. No sugar would mean the yeast is unable to respire. Respiring yeast produces alcohol, so why don't people get drunk when eating bread? When the bread is baked, the yeast is killed and the ethanol evaporates. Can evaporating ethanol help you think of another use for yeast? Yeast is also used in the production of beers and wines. This time, useful waste material is ethanol. To make beer, barley grains are soaked in water to cause them to germinate. This is known as malting. The malted grain is ground, which activates enzymes that turn the starches in the grain into sugars. The mashed barley is then boiled, and hops are added to give flavor. As the mixture cools, yeast is added. The yeast fungus consumes the sugars from the grains and starts to produce carbon dioxide and alcohol. The beer is usually filtered and the yeast is recycled to make more beer. So we've had yeast to make bread, and barley hops and yeast to making beer. What about wine? Wine can be made at home in a demijohn. Why do you think bubbles are visible? Why do you think there is an airlock in the neck of the demijohn? Pause the video to discuss this. The bubbles show that carbon dioxide is being produced as the yeast respires anaerobically. The airlock has two purposes. It allows the carbon dioxide out of the demijohn so that it doesn't explode. It also prevents air from entering the demijohn, ensuring the yeast continues to respire anaerobically and is not contaminated by other microorganisms. Hopefully, you now agree that yeast is a very beneficial microorganism that humans have learnt to use to our advantage, not only as mushrooms on our pizzas, but for making bread, beer, and wine.
For humans, food and drink are more than just a necessity for survival. They're a huge part of our culture and our history, shaping our lives on a daily basis. And alcohol has perhaps had the strongest influence on us, pun intended. From soft alcohols like beers and wines to hard liquors like whiskey and vodka, booze comes in many shapes and styles and can be found all over the world. But how did humans discover alcohol, and how is it made? Thanks to some microscopic organisms, some time, and some crafty chemistry, we've gotten pretty good at creating our own hooch. The alcohol we're describing today is ethanol, which is actually just one type of alcohol, but it's the only one that's safe to drink in large quantities because we've developed the ability to metabolize or break down and eliminate its toxic components. Ethanol occurs in nature thanks to fermentation, the process of breaking sugars down into ethanol and carbon dioxide. And humans and our non-human ancestors have been able to metabolize ethanol for a really long time. From examining our DNA, some scientists estimate that our ancestors developed the ability to metabolize ethanol around 10 million years ago, way before we were mixing our own cocktails. Back then, we hadn't even split off from the ancestors of chimpanzees, but that adaptation could have been helpful for our early primate ancestors by allowing them to eat fermented fruit without getting sick. Evolving humans must have enjoyed what they felt when they bacteria ate the fermented fruits, because figuring out how to create our own fermented beverages quickly became a centerpiece of human culture. In fact, there are some researchers who believe that our desire for alcohol is what drove us to develop agriculture, which would have given us constant access to the grains and starches needed to produce our own booze. The earliest known evidence of the deliberate creation of an alcoholic beverage is 9,000 years old, discovered in some pottery in the Yellow River Valley in China. This drink was some sort of wine and contained rice, honey, and grapes. In 2005, researchers actually recreated the drink based on a chemical analysis, and apparently it tasted, quote, very intriguing. Archaeologists think that around the same time people were originally making this stuff, barley beers and grape wines were being produced in the Middle East. People probably made those early drinks by leaving fruits and grains in covered containers for long periods of time. Eventually, they'd open the containers and the liquid inside would be alcoholic. But for thousands of years, no one knew exactly why this was happening. Thanks to modern science, we've got it pretty much figured out. Beers are produced by fermentation, which comes from the Latin fervere, meaning to boil. Which makes sense because the mixture of grains and water does bubble during the process. And a beer really only needs 4 basic components: grain, water, yeast, and hops. Different varieties of these components mixed in different ratios can give each beer a distinct flavor. There are also different ways to do the beer brewing itself, but here's how the process generally works. The first step in making beer involves cooking grains in water, which produces a mix called the mash. The heat activates enzymes within the grains and begins to break the starches down into simple sugars. Different grains are used for different kinds of beer. Dark roasted grains are used for darker, heavier beers like stouts, while light grains are used for light beers like wits and pilsners. Once they've all been cooked down, the grains are removed from the pot and the remaining liquid is boiled. At this point, the liquid is called the wort, and it's during this step that the primary flavoring element of the beer is added: hops. Hops are actually flowers, harvested from a climbing vine called Humulus lupulus. Beers have always been flavored with herbal mixtures, and we're not sure when or why hops became the herb of choice, but it was first noted as being part of brewing sometime in the 9th century CE. Adding hops to beer helped balance the sweet flavors from the grains with an acidic kick, but more importantly, the hops kept it from spoiling, so the beer lasted longer. We now know that hops produce antibacterial compounds called alpha and beta acids, which help keep the beer free of bacteria and support the growth and survival of the yeast. These acids mainly give beer its distinctive bitterness, but different varieties of hops can also give beers citrusy, floral, or even piney notes. After the boil, the liquid is rapidly cooled to prevent bacterial contamination and oxidation, which can change the taste of the beer. Once the wort is cooled, the yeast is added to begin the fermentation process. Yeast, in this case a particular variety called Saccharomyces cerevisiae, is a microscopic single-celled fungus. Yeast rely on sugars for energy, so they eat up the sugars in the wort and spit out carbon dioxide and ethanol as waste products. The carbon dioxide is why the liquid bubbles, and the ethanol is why there's alcohol in beer. During the first phase, the primary fermentation, there's a lot of yeast hard at work. All that waste carbon dioxide is allowed to escape the container, while the ethanol is left behind. After a while, usually a minimum of 2 weeks, most of the sugar has been consumed, and most of the yeast is dead. In some cases, the beer is transferred to bottles with a small amount of sugar to help fuel the remaining yeast through the last step: secondary fermentation. The beer is then tightly capped and the carbon dioxide and ethanol produced within the container are preserved, finally turning the liquid inside into the fizzy, intoxicating beverage that so many of us love. Wine production is similar to beer making in a lot of ways. It's just that instead of grains, wines use fruit to produce alcohol. And as you probably know if you've ever tasted a $3 bottle of wine, different varieties of grapes and how they're prepared can dramatically affect the flavor of the wine. First, the grapes are crushed. Back in the day, people used to stomp on the grapes in big tubs, but now it's generally done with specialized mechanical crushers. The resulting mixture of skins, pulp, and juices is called the must. Whether the wine is a red or a white usually depends on this step. White wines are often made from green grapes, and red wines are generally made from purple grapes, but the color of the grape doesn't automatically determine what type of wine it will be. In fact, purple grapes can be used to produce both kinds of wine. Leaving the dark grape skins sitting in the must will lead to a red wine, while separating the juices from the skins and pulp quickly will lead to a white wine. Those dark grape skins also contain tannins, compounds that a chemist would call polyphenols. They're made of lots of connected rings of carbons attached to an oxygen and a hydrogen. These compounds give red wine its characteristic dryness and help red wines age well, smoothing and mellowing the flavor over time. As with beer, the key component in turning grapes into wine is the presence of yeast. In the old days, naturally occurring wild yeast on the grapes, mashed into the must by winemakers' feet, would kickstart the process. These days, winemakers sterilize the must and add their own specially selected yeast for better control over the wine's flavor. And just like with the beer, the yeast converts the grape sugars into ethanol and carbon dioxide. After fermentation, fermentation, the yeast is killed off or filtered out, and the liquid is clarified to remove particles that might affect the taste or appearance of the wine. The wine may then be aged in wooden barrels for a few months to a few years, which gives the drink more flavors. Finally, it's bottled and shipped off for consumption, ready to be sipped by vinophiles everywhere. So these soft alcohols, as it turns out, are pretty easy to make by mistake. But what if you wanted to throw a really crazy party? How do we make hard liquor? High alcohol content beverages like vodka and whiskey require an additional step: distillation. Distillation is a process of deliberate evaporation, cooling, and condensation that produces purified liquids. Creating hard liquors this way takes advantage of the fact that ethanol has a lower boiling point than water, so it turns into a gas at a lower temperature than water does. Just like with beer and wine, hard liquors begin with a fermentation step. Different fruits, grains, and starches can be used to create this initial brew depending on the liquor. For example, potatoes are generally used for vodka, while bourbon is made from corn, rum comes from sugarcane, and tequila is made from agave. The yeast gobbles up all of those sugars and produces ethanol and carbon dioxide, but the liquid has a relatively low alcohol content, around 10 to 15%. So, to make the liquor truly hard, we have to purify some of that liquid to give it a higher alcohol content. The liquid is transferred to the still, where it's heated inside the pot until the components with lower boiling points begin to boil off. As the solution vaporizes, the temperature is increased very slowly, pushing the other compounds to boil at the appropriate temperatures. The boiled off vapor rises to the top of the pot and passes into the distillation column, where specialized plates cool the vapor and cause it to condense and drop back into the pot. This gives distillers precise control over the spirits as they pass up through the column, allowing them to make adjustments to the contents and flavor of the drink. The compounds with the lowest boiling point are able to reach the top of the column and pass into the line arm, a horizontal pipe that condenses the vapor back into liquid so it can be collected. This distillate is collected in stages, with each stage kept separate by its boiling points. The first portion of the distillate, called the heads, contains the most volatile compounds, the liquids with the lowest boiling point. These include acetone and esters, and small amounts of these compounds are saved to add flavor to the final product. The second portion of the distillate is called the heart, and it's the most important part because it contains all of that intoxicating ethanol. The heart is collected, mixed with some of the heads, and usually further modified until it reaches the desired flavor profile, which sounds really strange out of context. Some of these modifications involve flavoring the liquid directly, like by adding juniper berries to gin. They can also involve redistillation, passing the distillate through the process all over again. And of course, hard liquors can be modified through aging. Like wine, the type of container a spirit is aged in can have a significant effect on its behavior. Bourbon is aged in newly cut and freshly charred oak barrels, imparting a rich, smoky flavor to the drink. Scotch, on the other hand, is aged in used barrels, usually in barrels that have been previously used for aging bourbon or sherry. Different varieties of the same kind of spirit can age for different amounts of time too, like how white or blanco tequila isn't aged, but reposado or rested tequilas have been aged in oak for at least 2 months. So there are a lot of ways to turn that distilled ethanol into many different kinds of liquors available around the world. Scientists are working in breweries and distilleries every day to combine these scientific techniques in new and interesting ways, creating super bitter beers and unusual kinds of liquors. And all of these beverages are part of our culture and our history, from the earliest beers to the latest developments in distillation technology. Thanks for watching this episode of SciShow, brought to you by our patrons on Patreon.
Now we'll just briefly talk about methanogens. Methanogenesis occurs in the domain Archaea. These organisms are found in soil, also in digestive tracts of ruminants. These are areas where there is no oxygen available, and this is an important process of decomposition. In methanogenesis, these organisms do not use oxygen as the final electron acceptor, very similar to fermentation in lactic acid and alcohol fermentation. The pathways of methanogenesis are quite a bit more varied than the pathways that we've seen so far, so we're not going to go into all the details of them. Some of them will use carbon dioxide as that final electron acceptor and also produce methane. As we said, there's a variety of pathways, but we'll just look at a couple here. In some pathways, we take carbon dioxide at the end and we add hydrogen producing methane and water. And then sometimes we will take this hydrocarbon and produce methane and carbon dioxide. So it just depends on the pathway. And again, these are only a few of the pathways. There's many more pathways that methanogens can use. Here we'll take a look at a video that's related to methanogenesis. It's interesting to note that even though methanogenesis has been around forever, the domain archaea were the first living organisms, we still don't understand the process completely. And it's probably because it does have a variety of these different pathways. It's also a very important process in decomposition. As the video showed us, there's a lot to be learned in how these microorganisms interact, not only themselves in producing methane, but how they interact with one another in somewhat symbiotic relationships. Now we're going to take a look at another type of anaerobic pathway called anaerobic cellular respiration. So it's a little bit confusing because we have anaerobic pathways. Glycolysis technically is anaerobic. Lactic acid fermentation, alcohol fermentation, methanogenesis, those were all anaerobic as well. But the term anaerobic cellular respiration is used in a very special way. It's not just an anaerobic pathway. When you put all three of those words together and you say anaerobic cellular respiration, you're really looking at this pathway that we see here in the middle. Over on the far left, we have aerobic respiration. Aerobic cellular respiration, we had glycolysis, we went through the citric acid cycle, we did electron transport, we used oxygen as our final electron acceptor. Over here in fermentation, it was still anaerobic because we didn't use oxygen, but we also didn't go all the way through the complete breakdown of the sugar. We ended up with either lactic acid or alcohols. Here in the middle, we have anaerobic cellular respiration. In anaerobic cellular respiration, it's going to look a lot like aerobic respiration. It's going to go through all of the pathways. Glycolysis hasn't really changed. The citric acid cycle hasn't changed. We still do electron transport. The big difference here is just what serves at the very end as that final electron acceptor. It's not oxygen here because oxygen is not available in this anaerobic condition. Here we have different electron acceptors. It could be sulfate ions, nitrate ions, or carbonate ions, or sometimes it can just be sulfur. So let's take a look at anaerobic cellular respiration. This is used by some bacteria, some archaea, and it can be done either obligately or facultatively. What that means is if you're an obligate anaerobe, you cannot do cellular respiration in the presence of oxygen. Basically, oxygen will kill you. And a facultative anaerobe is one that could be anaerobic or could be aerobic. These organisms, some of them are obligate and they have to be in anaerobic conditions. Others can switch over to anaerobic if the need arises. As we said, instead of oxygen being that final electron acceptor, it is some other type of a molecule. It may be nitrate, it may be sulfate, it may be sulfur, or we saw on the previous slide, it could be carbonate. We're looking at pretty much the same type of pathway. We're still making ATP using the proton motive force. We're still breaking down glucose and producing carbon dioxide, but instead of it being aerobic in the presence of oxygen, here in anaerobic, we're going to have other electron acceptors. This shows us a little bit more detail. Up at the top, we have aerobic respiration, where oxygen was that final electron acceptor at the end of the transport chain. And down here, we just have slightly different transport chain, where instead of it being oxygen, we have nitrate as the final electron acceptor. When these organisms perform anaerobic cellular respiration, then the ATP gain is very similar to what it would be in cellular respiration. It's not always identical, but it's certainly much higher than those that do fermentation that only get the 2 ATP from glycolysis, and everything else is about regenerating NAD. That's not the case here. There's quite a bit of ATP produced in anaerobic cellular respiration. So that concludes our section on what organisms do in the absence of oxygen. Next, we'll take a look at what they do when there perhaps is not enough glucose available.